Welcome to the deep dive. We are skipping the pleasantries today because we have a mountain of utterly fascinating, highly technical material to get through, and I really just want to jump straight into the data. Our mission for this deep dive is to explore the hidden, constantly shifting world of mushrooms.
Right. And we are going far beyond standard foraging profiles today.
Exactly. We're looking at the foundational architecture of how we name these organisms, how we map their incredibly complex and, frankly, often deceptive family trees, and then looking at the clinical side, how our individual enzymatic and digestive systems react to them in wildly unpredictable ways.
Yeah, we are talking about microscopic deception, genetic cold cases hidden in dusty museum archives, and physiological betrayals by universally praised culinary species.
That's a lot. And to do this, we are pulling from a dense stack of literature. First, we have a massive 2024 taxonomic study from the journal Mycosphere. This one radically reorganizes the Omphalotaceae family, with this massive data set drawn from new discoveries in China.
It's just a huge structural shift.
Huge. Second, we are digging into a pivotal 2010 Mycology paper, and that's supported by an in-depth profile from mushroomexpert.com. That one essentially exiles a very specific, uniquely clustered mushroom into a brand new genus of its own.
And finally, we are analyzing a comprehensive toxicological report from MycoWeb that details the specific biochemical and physiological reasons why perfectly edible, highly sought-after mushrooms trigger intense gastrointestinal distress in seemingly random individuals.
Okay, let's unpack this. What happens when the morphological traits of a macrofungus completely contradict its phylogeny? And what happens when a widely celebrated edible mushroom fundamentally clashes with your specific, highly individualized gut chemistry?
The through line connecting both of these phenomena is really the stark limitation of human observation when it's applied to fungal ecology. Because for the entire history of classical taxonomy, we relied heavily on macroscopic and microscopic morphology.
What we can actually see.
Right. What we can observe, measure, and stain just to construct these neat evolutionary narratives. But the fungal kingdom operates under intense environmental pressures that frequently result in extreme morphological convergence. What we're seeing now with the maturation of molecular phylogenetics is a necessary, albeit chaotic, period of tearing up the old maps.
And on the toxicological front, the literature reveals that even when the taxonomy is perfectly resolved, the unique biochemical fingerprint of the human microbiome and, our enzymatic pathways proves that our relationship with these organisms is never a static variable.
Never.
So tearing up the old maps is exactly what that 2024 Mycosphere paper is doing. And it brings us to the first major segment of our analysis, the Great Fungal Family Tree Shakeup.
It really is a shakeup.
Looking at the data, the taxonomic battlefield centers heavily on the family Omphalotaceae, which is typified by the genus Omphalotus. If you look at the historical literature, the methodology was entirely phonetic. You had early mycologists categorizing specimens based purely on gross morphology.
Yeah, you looked at the pileus, the stipe, the lamellar attachment, the spore print.
Right. But our sources highlight that this purely morphological approach has created a systemic phylogenetic disaster, particularly when you look at the genus Gymnopus.
The Gymnopus problem is a textbook example of rampant polyphily. When we map out the genetic lineages, we find that the traditional boundaries of Gymnopus encompassed multiple, entirely distinct evolutionary clades that do not share an immediate common ancestor.
So it's a massive polyphyletic group.
Exactly. The best way to understand polyphily is imagine finding out that the people you thought were your siblings just happen to look exactly like you, but they actually have entirely different parents.
Wow.
That is exactly what happened to this massive genus. The issue stems from the fact that the colybioid and marasmioid fruiting body forms...
That classic, tough, thin-fleshed cap and wiry stem.
Right. That physical structure is a highly successful functional adaptation. It's a morphological strategy for dealing with specific microenvironmental stressors like rapid desiccation. And because that form is so functionally effective, completely unrelated genetic lineages have independently converged on the exact same physical architecture.
So historical taxonomists were grouping these fungi based on functional convergence rather than actual genetic relatedness.
Yes.
It is incredible to think about the sheer scale of convergent evolution required for completely different branches of the phylogenetic tree to produce fruiting bodies that are virtually indistinguishable to the naked eye. I mean, the only way this structural deception was exposed was through the systematic application of molecular markers.
The modern methodology is entirely reliant on those markers.
The Mycosphere paper heavily leverages the internal transcribed spacer, the ITS region, alongside the nuclear large subunit, the LSU. And it makes sense given that the flanking ribosomal RNA genes are highly conserved, while the spacer regions evolve fast enough to give us that species-level and genus-level resolution.
The reliance on the ITS and LSU regions has become the gold standard precisely because of that balance between conservation and variability. Because fungal morphology is notoriously plastic. A single species can exhibit radically different phenotypical expressions depending on the substrate composition or ambient humidity and temperature during primordia formation.
It can look completely different on Tuesday than it does on Friday.
Exactly. But the sequence of the ITS region cuts through that phenotypic noise. By utilizing these molecular barcodes, researchers can quantify the actual phylogenetic distance between specimens. When the morphometrics suggest two specimens are closely related, but the ITS alignment shows massive divergence, the molecular data takes precedence.
It forces a taxonomic realignment regardless of how visually similar the specimens are.
It absolutely does.
And when you look at the methodology section of the 2024 study, the sheer volume of data they processed is staggering. We are not talking about a localized sampling effort here.
Yeah.
The researchers generated and analyzed 387 ITS sequences and 269 LSU sequences.
The taxonomic fallout from processing that much molecular data is immense.
They formally described 15 entirely new species, erected a completely novel genus, and proposed 9 new combinations. Those 9 new combinations represent existing, well-documented species that had to be forcibly evicted from their historical genera because the DNA proved they had been misclassified for decades.
If we connect this to the bigger picture, this study represents a systemic audit of biological diversity. The broad morphological concept of Gymnopus enceladus has been undergoing continuous, aggressive deconstruction since the late 1990s.
Right.
The introduction of novel genera to accommodate these orphaned clades isn't just academic housekeeping. It is a fundamental correction of our evolutionary models. Science is actively redrawing the map of the natural world. We are reallocating taxa to ensure that our nomenclature actually mirrors the phylogenetic reality rather than perpetuating historical errors based on superficial morphological similarities.
That concept of correcting historical errors leads perfectly into one of the most compelling narratives in the Mycosphere paper, something we can frame as the 100-year-old mystery of Gymnopus contrarius.
This is a great story.
It perfectly encapsulates the chaos of morphological taxonomy. So if we go back to 1911, Charles Horton Peck, a massively influential figure in early American mycology, described a specimen collected in New York State. The ecological notes place it in association with moss, Phaceae species, so spruce, and Abies balsamea, balsam fir.
And Peck originally published it as Marasmius contrarius.
Right. But tracing this specific taxon through the literature over the next century reveals a complete lack of consensus among taxonomists.
The specimen became a perennial outlier in the literature. Because its suite of morphological characters defied easy categorization, subsequent taxonomists continually reassigned it in an attempt to find a logical phylogenetic home.
It kept getting bounced around.
Bounced around for a century. It was transferred out of Marasmius and moved into Colibia, where it languished under the name Colibia umbonatella. Eventually, as the boundaries of Colibia were restricted, it was shunted over to Gymnopus as Gymnopus contrarius.
But the DNA didn't agree.
Not at all. Molecular analysis consistently demonstrated that it was an anomaly. On every phylogenetic tree constructed for the Omphalotaceae, this specific lineage formed an ill-defined monotypic clade.
Meaning it was on a branch of the family tree all by itself.
Entirely on its own. It occupied a highly isolated long branch, exhibiting no significant statistical support linking it to any of the established subclades within the Gymnopus sensu stricto complex.
It's essentially a taxonomic orphan. You have this century-old historical type specimen, designated NYSF 859, resting in the New York State Museum archives. For decades, it's just a dried husk in a cabinet, representing a monotypic lineage that defies placement.
But the researchers in the 2024 study didn't just leave it as an anomaly. They applied a highly advanced methodological approach to extract viable data from it. They used genome skimming via the Illumina Novasec 6000 platform.
Which is incredible technology.
It is. When you are dealing with a type specimen from 1911, traditional Sanger sequencing of specific loci often fails completely because the DNA is highly degraded and fragmented.
So how does genome skimming bypass that?
The application of genome skimming in this context is a brilliant utilization of modern bioinformatic pipelines. With historical herbarium specimens, the genomic material is typically sheared into very short fragments due to decades of desiccation and chemical degradation. Attempting targeted PCR amplification of the ITS or LSU regions is frequently impossible because the necessary primer binding sites are destroyed.
The targets are just gone.
Exactly.
Yeah.
So genome skimming circumvents this limitation. Instead of targeting specific markers, the Illumina platform performs low-coverage, shotgun sequencing across whatever genomic fragments remain in the sample.
It just reads everything it can find.
Right. By generating millions of these short reads, researchers can use computational assembly algorithms to reconstruct the high copy number regions, specifically the ribosomal RNA operon, which contains the ITS and LSU sequences. It is an incredibly powerful technique for recovering phylogenetic data from compromised century-old material.
And the resulting phylogenetic tree from that genome skimming data provides absolute clarity. The molecular data unequivocally proved that this lineage was so deeply divergent that retaining it within Gymnopus, or any previously described genus, was taxonomically indefensible. It required the erection of a novel genus,
And they named it Peckorumyces.
combining the original discoverer Peck's name with myces for fungus, establishing the current binomial as Peckorumyces umbonatus.
What makes the Peckorumyces resolution so critical is its placement in the broader topology of the family tree. The multi-gene phylogenetic analysis demonstrated with a robust statistical support that Peckorumyces does not even fall within the primary Gymnopus clade.
Where did it land?
It resolves as a sister clade to the genus Anthracophyllum. And this is a profound illustration of morphological deception. Anthracophyllum species are typically characterized by pleurotoid or crepidatoid fruiting bodies. They often lack a distinct stipe and grow directly from wood.
Which is morphologically alien to the classic stipitate colibioid form of Peckorumyces umbonatus.
Exactly. Yet genetically, they share a much more recent common ancestor with each other than Peckorumyces does with any species of Gymnopus.
It forces a complete paradigm shift in how we interpret physical form. And for you listening, considering the broader implications, this completely recontextualizes the function of museum archives. These herbarium collections are not static repositories of settled science. They are active biological cold cases waiting for technology to catch up.
They are massive vaults of unresolved biological data.
You have tens of thousands of type specimens cataloged under legacy names. The Peckorumyces study proves that as high-throughput sequencing and genome skimming technologies become more accessible, we're going to see a tidal wave of historical specimens systematically reevaluated and reclassified. The archives are actively generating novel evolutionary data.
The archival reassessment is going to be a defining characteristic of mycological taxonomy for the next several decades. We are transitioning from a period of morphological assumption to a period of strict molecular auditing.
This transition isn't just happening with century-old specimens in New York either. The bulk of the 2024 Mycosphere paper focuses on active contemporary fieldwork, specifically mapping out the hyperdiverse ecosystems in China. Let's take a tour of China's strange new fungi.
There's an immense amount of diversity detailed here.
The study introduces 15 newly discovered species, and what immediately stands out in the ecological data is the extreme geographic and environmental specificity driving this speciation. You can't just find these anywhere.
The high rate of novel discoveries in these regions is deeply tied to the varied topography and the resulting microclimates. Fungal populations in mountainous regions frequently experience intense isolation.
Get cut off.
Right. When a population is confined to a specific elevational band, associating with a highly restricted set of host plants under unique climatological pressures, the gene flow with other populations is severed. This allopatric isolation accelerates genetic divergence.
So they evolve rapidly in a vacuum.
The unique morphometrics and chemical profiles documented in these new Chinese species are direct evolutionary responses to those highly specific environmental pressures.
Let's drill down into the specific data on a few of these novel taxa, starting with Connopus alpina. The collection data indicates this species was discovered in Yunnan province at an extreme elevation of 3,937 meters. That is staggering.
The environmental stressors at nearly 4,000 meters are severe.
Intense ultraviolet radiation, significant diurnal temperature fluctuations, and low atmospheric pressure. Macroscopically, it presents with a distinctively dark brown pileus. It's tiny. But the microscopic data is where the specific adaptations become apparent.
The cytological dimensions are highly diagnostic for Connopus alpina. The researchers noted exceptionally long basidia, measuring between 39.9 and 54.3 micrometers.
And the basidia are the spore-producing structures.
Correct. In the context of related Polyphyletic species, this elongation is a significant morphological deviation. While the exact functional advantage requires further physiological study, modifications to basidial architecture are often linked to optimized sporulation mechanics under specific atmospheric conditions.
And what about that dark coloration?
The dense, dark pigmentation of the Polyphyletic is a classic adaptation observed in alpine fungi. The synthesis of complex melanin compounds provides critical shielding against the high levels of UV radiation present at that altitude. It protects the underlying hymenium and developing basidiospores from fatal DNA damage.
A built-in sunshield. Moving from the alpine extremes of Yunnan, the study describes a fascinating collection from Jilin province, Genopus chowi.
This one has a great backstory.
The geographical notes are highly specific. It was collected in close proximity to a statue honoring Chongquang Chao, also known as Zhonghuangzu, a foundational figure in Chinese mycological research.
And while naming the species in his honor is a great historical nod, the field notes detail a highly distinctive characteristic that makes this species immediately recognizable in the field.
It possesses a powerful, pervasive odor of rotten cabbage.
Yes, the analysis of volatile organic compounds, or VOCs, in macrofungi is a deeply complex area of study. While a rotten cabbage odor might seem like a mere taxonomic curiosity, these secondary metabolites serve highly specific ecological functions.
They aren't just smelling bad for no reason.
No. The production of sulfurous compounds, such as dimethyl sulfide or dimethyl disulfide, which frequently mimic the scent of decaying brassicas or carrion, is genetically encoded. In many cases, these specific VOC profiles evolve to attract very specific dipteran or coleopteran species.
Flies and beetles.
Right. Insects that utilize the fruiting body for oviposition, inadvertently acting as highly effective vectors for spore dispersal. Conversely, the specific chemical cocktail might serve as a potent deterrent against local alpine fungivores.
So it's either ringing the dinner bell for bugs that will spread its spores, or it's chemical warfare to stop things from eating it.
From a strictly taxonomic perspective, the presence of this specific genetically fixed VOC profile provides a crucial diagnostic tool in the field. It cleanly separates Gymnopus chowi from sympatric, morphologically similar species before any molecular work is even initiated.
The integration of sensory data with molecular phylogeny is a vital part of field taxonomy. But occasionally, the unique chemistry of a species isn't apparent until you expose it to specific reagents in the lab. This brings us to Gymnopus fuscus,
Another high-altitude specialist.
discovered in Qinghai province at an astounding 4,069 meters. The specific epithet fuscus refers to the dark brown coloration of the pileus. However, the micromorphological analysis revealed a bizarre chemical reaction.
The application of strong alkaline reagents, specifically potassium hydroxide, or KOH, is a standard procedure when preparing slide mounts for microscopy. It rehydrates tissue and often clarifies cellular structures.
But with Gymnopus fuscus, something else happens.
The introduction of KOH triggers a dramatic and highly specific chromatic shift. The researchers documented that specific microscopic structures, namely the basidioles, the chylocystidia located on the lamellar edges, and the terminal cells comprising the pyapellis, all turn a distinct green color when mounted in KOH.
Just a sudden shift to green under the microscope.
This colorimetric reaction indicates the presence of a very specific localized class of secondary metabolites. It's likely a unique anthraquinone or complex phenolic compound embedded within the cell walls of those specific structures.
And because this chemical reaction is absent in its closest phylogenetic relatives, the KOH green schist serves as an absolute, definitive microscopic barcode for identifying Gymnopus fuscus.
It highlights how critical thorough microscopic and chemical analysis remains even in the era of ITS sequencing.
Absolutely. Rounding out the highlight reel from the Chinese study, we have Gymnopus niveus, collected in Hunan province. Macroscopically, it stands out due to its pure snowy white coloration and a deeply sulcate or grooved pileus margin. The molecular data cleanly separates it, but the structural arrangement of the cap cuticle is also highly distinct.
The architecture of the Polyphyletic in Gymnopus niveus is fascinating. It is organized as a trichoderm, meaning the outermost hyphae are oriented somewhat perpendicularly to the surface of the cap, rather than running parallel as in Acutus.
And there's more to it than just the orientation.
Yes, the data details that these terminal hyphae are highly diverticulate. They are covered in numerous small, peg-like or coralloid projections. This specific micromorphology significantly increases the total surface area of the Polyphyletic at a microscopic level.
Which changes how it interacts with the environment.
In certain environmental contexts, this diverticulate trichoderm structure can profoundly influence how the fruiting body interacts with ambient moisture, potentially altering evaporation rates or facilitating the condensation of dew in its specific microhabitat.
It is a perfect example of how macro-level appearance, like that deeply grooved snowy cap, is intrinsically linked to intricate micro-level cellular engineering. Every single newly sequenced species from this study reinforces the same conclusion.
The sheer volume of cryptic diversity hidden within these isolated microclimates is staggering.
What we previously assumed were minor regional variations of widespread species are, upon molecular inspection, entirely distinct evolutionary lineages with highly specialized biochemical and structural adaptations.
We are just barely scratching the surface of what is out there.
Now, shifting our focus from the broad taxonomic surveys in Asia, I want to drill down into a highly specific phylogenetic conflict outlined in our North American sources. We are transitioning to the 2010 Mycology paper authored by Hughes and collaborators alongside the detailed taxonomic breakdown from Mushroom Expert.
This is a classic case study.
Here's where it gets really interesting, because we are analyzing a scenario where the molecular data doesn't just shuffle a species between existing genera. It forces the creation of a new genus for a fungus that is arguably one of the most easily recognized species in the Northern Hemisphere. We are looking at the taxon historically literature-bound as Gymnopus acervatus, or in older texts, Polyphyletic acervata.
The acervatus complex represents one of the most compelling case studies in the friction between deeply entrenched field taxonomy and modern molecular phylogenetics.
People were very confident about this one.
Extremely confident. For decades, morphological taxonomists and field guide authors across North America and Europe considered this taxon to be a thoroughly resolved, cleanly defined species, perfectly situated within the broader Gymnopus or Polyphyletic frameworks. Its macroscopic profile is so consistent and distinct that it rarely presented identification challenges in the field.
I mean, let's describe it vividly so you listening can visualize it. If you are surveying a mixed conifer forest, particularly in late summer or fall, the ecology of this species is highly specific. It is saprobic, growing almost exclusively on the decaying wood of conifers, frequently emerging from stumps or buried logs.
And it grows in these dense, tight clusters.
Right, the fruiting habit is densely cespitose. You find these massive, tightly packed clusters of fruiting bodies. The cap is convex, lacking the distinct umbo you see in related species, and exhibits a striking, rich, reddish-brown coloration that is highly hygrophilous, meaning it fades dramatically to a pale pinkish buff as it loses moisture.
The stipe shares that reddish-brown tone and is notably hollow.
But the key diagnostic feature, the morphological anchor for this species, is that the stipe is completely glabrous. It is entirely smooth and bald, devoid of any fuzz or texture, except for a little bit of whitish fuzz, that conspicuous accumulation of basal mycelium where it directly interfaces with the wood.
And white spore print. That specific combination of characters, the dense cespitose growth on conifer wood, the hygrophilous reddish-brown pileus, and crucially, that entirely glabrous stipe with the white basal mycelium created a morphological consensus. It was universally accepted as a core member of the Gymnopus complex.
But then the DNA testing happened.
When the Hughes team subjected the Gymnopus and Rhodocolibia genera to rigorous multi-gene phylogenetic analysis targeting both the ITS and LSU regions, the molecular data completely dismantled that morphological consensus.
The resulting phylogenetic tree is essentially a document of genetic eviction. Looking at the topology presented in the 2010 paper, the placement of the acervatus sequence data is jarring. It doesn't nest within the primary Gymnopus clade.
Nor does it align with Rhodocolibia.
Instead, the acervatus lineage forms a massive, supportless long branch. What does that actually mean on a phylogenetic tree?
The presence of a supportless long branch in a robust phylogenetic analysis is highly significant. In a well-sampled tree, related species cluster together on shorter branches, indicating relatively recent divergence from a shared common ancestor.
So they're close cousins.
Right. When a sequence resolves on a long branch that lacks statistical support linking it to any surrounding clades, it indicates a deep, ancient evolutionary divergence. The lack of support essentially means the molecular algorithms cannot confidently connect it to any extant sister taxa in the dataset.
There's an island.
It is an evolutionary island. The genetic distance between the acervatus collections and the true Gymnopus species was so vast that retaining it within that genus would have rendered the entire genus paraphyletic and taxonomically meaningless. The DNA was unequivocal. This morphologically classic Gymnopus was an entirely independent evolutionary lineage.
So to resolve this phylogenetic incongruence, the Hughes team formally erected the novel Genus Connopus, specifically to accommodate this single, highly divergent lineage. The densely clustered reddish-brown conifer specialist was officially recombined as Connopus acervatus.
But the analysis in the Mycology paper goes significantly deeper than just genus-level realignment.
And this is a great teaser for the future of taxonomy. By analyzing multiple collections of Connopus acervatus from across its Holarctic distribution, the researchers uncovered a layer of cryptic genetic diversity that points directly toward active allopatric speciation.
The geographic resolution within the molecular data is arguably the most forward-looking aspect of the study. The researchers analyzed IT sequences from specimens collected in Western North America, Eastern North America, and Scandinavia.
And morphologically, they all look exactly the same.
Identical. Macroscopically, a specimen from the Pacific Northwest looks utterly identical to a specimen from New York or Sweden. They all exhibit the exact same morphometrics, the same cespitose growth, the same glabrous stipe.
But the sequence alignments revealed a distinct phylogenetic split.
The collections from Eastern North America and Scandinavia clustered together with high support. That indicates a contiguous genetic lineage that likely maintains connectivity, perhaps via historical circumboreal pathways.
However, the sequences isolated from the Western North American collections formed a completely distinct divergent subclade.
Exactly. The genetic divergence of the Western North American clade is substantial enough to strongly suggest reproductive isolation. This implies that massive geographical features, specifically the continuous barrier of the Rocky Mountains and the arid expanse of the Great Plains, have effectively severed gene flow between the Western and Eastern populations.
We are literally observing the molecular mechanics of cryptic speciation.
While stabilizing selection has maintained the highly successful macroscopic morphology uniformly across the continent, genetic drift and localized mutations are accumulating in isolation. The 2010 analysis posits that as more robust multi-locus data becomes available, the Western North American clade of Connopus acervatus will likely need to be recognized and formally described as a completely separate biological species.
It is a profound realization that the morphological uniformity we observe in the field can completely mask deep, ongoing evolutionary divergence.
The outward form remains the same, but the internal blueprint is slowly rewriting itself.
This tension between what we observe and what is actually happening at a molecular level brings us to the final and arguably most complex segment of our analysis. We spent the first half of this deep dive establishing how precisely molecular taxonomists can map the genetic lineage of a fungus, placing it on the exact right branch of the phylogenetic tree.
But there is a very human element to all of this.
Exactly. Bridging taxonomy and digestion. What happens when you perfectly identify a universally recognized, highly prized edible mushroom, verify its taxonomy beyond any doubt, prepare it correctly, and your specific physiological system violently rejects it?
This transition moves us from the complexities of fungal phylogenetics into the deeply unpredictable realm of human toxicology, specifically focusing on the phenomenon of idiosyncratic gastrointestinal toxicity.
We are pulling directly from the comprehensive toxicological reports compiled by MycoWeb. These detail exactly why the concept of edibility is not a universal biological constant.
It's a spectrum, really.
When we discuss idiosyncratic toxicity, we are not talking about accidental ingestion of cyclopeptide-containing species like the death cap Amanita phalloides. We are analyzing cases where a known, safe, widely consumed culinary species triggers severe physiological distress in a highly specific subset of the population.
And the literature highlights a fascinating historical case involving Alexander H. Smith,
one of the most preeminent mycologists of the 20th century. This story perfectly isolates the idiosyncratic variable. Tell us about the fairy ring.
Alexander H. Smith's expertise is central to the validity of this case study. As a dominant figure in North American fungal taxonomy, his ability to accurately identify species in the field was absolute. The possibility of misidentification in this scenario is effectively zero.
Right, he literally wrote the book on many of these species. So the report details an event in 1958 at the University of Michigan. Smith harvested a substantial quantity of Lepista irina, which is a species universally recognized as a choice edible, and he found it in a naturally occurring fairy ring.
He meticulously verified the identification, confirming the macroscopic and microscopic features. He then served these verified specimens. Now, the crucial biological detail here is the origin of the mushrooms, a single fairy ring.
Why does that matter so much?
The fairy ring origin is the critical control variable in this natural experiment. A fairy ring is the fruiting expression of a single, continuous, radially expanding mycelial network. Therefore, every individual fruiting body harvested from that specific ring is a clone.
They are genetically identical.
They share identical genetics and consequently an identical biochemical profile.
Hmm.
There is no variance in secondary metabolites or protein structures between the specimens.
So Smith serves these genetically and biochemically identical, expertly verified edible mushrooms to a group. Smith himself consumes them without issue, as he had for years. His students consume them without issue. However, one specific husband and wife in the group experienced a severe, rapid-onset gastrointestinal crisis.
Intense nausea, vomiting, severe cramping. They woke up in the middle of the night.
They consumed the exact same biochemical compounds as everyone else, yet their bodies reacted as if they had ingested a potent toxin.
This raises an important question regarding the highly individualized nature of human digestion and immunology. In true idiosyncratic reactions, the fungal tissue itself does not contain universally toxic compounds.
It's safe food for almost everyone.
The physiological crisis is triggered by a fundamental incompatibility between the complex array of fungal secondary metabolites, novel proteins, and structural carbohydrates, and the unique enzymatic and microbiome profile of the individual consumer.
It's a mismatch.
Exactly. Fungal cell walls are heavily fortified with chitin, and the tissue contains a massive diversity of compounds not found in plant or animal food sources. If an individual harbors a specific genetic downregulation of certain digestive enzymes, such as trehalase deficiency, which severely impairs the breakdown of the fungal sugar trehalose, or if their gut microbiome lacks the specific bacterial consortia required to process these complex molecules, the accumulation of undigested compounds acts as a severe osmotic irritant in the lower GI tract.
So your body just can't break it down, and it rebels.
Alternatively, the reaction can be immunological. The gut-associated lymphoid tissue erroneously flags a harmless fungal protein as a hostile pathogen, triggering a massive, localized inflammatory purge response.
It essentially functions as a hyperspecific, highly localized physiological rejection. It's Russian roulette in the frying pan. And when you review the toxicology reports, there's a consistent list of usual suspects. Species that are aggressively foraged and highly valued, yet are statistically notorious for triggering these idiosyncratic cascades.
The first major complex the data addresses is the widely consumed chicken of the woods, the Laetoporus genus.
The Laetoporus complex requires careful analysis because the historical taxonomy masks significant underlying diversity that directly impacts toxicology. For decades, older field guides applied the binomial Laetoporus sulphureus as a catch-all for any large, yellow-pored, shelving polypore.
But modern mating studies and molecular data have proven that true Laetoporus sulphureus does not occur in Western North America.
Right. In regions like California or the Pacific Northwest, foragers are interacting with distinct genetic clades. Specifically, specimens fruiting on angiosperms, predominantly eucalyptus or oaks, are identified as Laetoporus gilbertsoni. Conversely, those restricted to gymnosperm substrates, primarily old-growth conifers, are Laetoporus conifericola.
The distinction between these species is highly relevant to the clinical data. The 1975 report from the San Francisco Toxicology Committee presents a compelling dataset. Out of 158 investigated fungal poisoning reports, they isolated 7 distinct cases involving severe, rapid-onset reactions specifically linked to Laetoporus ingestion.
And the clinical presentation is intense. Rapid onset of severe nausea and vomiting, frequently occurring within 5 to 45 minutes of ingestion, followed by a relatively rapid resolution over a few hours.
The rapid onset of symptoms is a key clinical indicator. Initially, the mycological community hypothesized that substrate toxicity was the primary driver. Because Laetoporus gilbertsoni frequently parasitizes eucalyptus, it was theorized that the mycelium was absorbing and concentrating toxic essential oils or complex phenols from the host tree.
Make the mushroom itself chemically volatile.
Right. However, expanded epidemiological data demonstrated identical rapid onset crises resulting from Laetoporus gilbertsoni harvested from oaks and, crucially, from Laetoporus conifericola harvested from pines.
So it wasn't the tree.
While host-derived compounds might contribute minor variants to the chemical profile, the data firmly establishes that the primary drivers of Laetoporus toxicity are twofold. First, the inherent idiosyncratic sensitivity of the individual's enzymatic pathways to the specific complex carbohydrates present in the Laetoporus tissue, and second, the absolute necessity of thermal degradation.
The thermal degradation aspect is critical. The reports emphasize that consuming Laetoporus raw, or even slightly undercooked, massively increases the statistical probability of a severe GI event. The chitinous matrix and the complex secondary metabolites must be subjected to prolonged heat to begin breaking down those molecular bonds before they enter the human digestive tract.
You have to cook it thoroughly.
If we move from polypores to what is arguably the most globally recognized premium wild edible, we have to look at the data on chanterelles. The Cantharellus genus is universally considered a premium edible, yet the reports document a striking case of hyperspecific intolerance.
The clinical literature contains a highly illuminating case report of an individual who developed a profound, idiosyncratic intolerance specifically to the European chanterelle, Cantharellus acervatus.
This poor guy.
The subject experienced severe, immediate nausea and vomiting after consuming a standard culinary preparation. Assuming this was an isolated incident of foodborne illness or improper handling, the subject later consumed a commercially prepared soup in Switzerland. The menu did not list mushrooms, yet the subject experienced the exact same rapid, violent physiological rejection.
And it turned out.
Subsequent investigation revealed the kitchen utilized a minuscule quantity of Cantharellus acervatus exclusively for aromatic flavoring.
The cook secretly added a tiny bit just for flavor. The fact that a microscopic quantity, used merely for aroma, could trigger such a massive physiological response demonstrates the extreme sensitivity of these idiosyncratic pathways. Your immune system, or enzymatic receptors, essentially memorize the specific molecular signature of that fungus and treat it as a critical threat.
And crucially, the taxonomic data intersects with the clinical data here again. While the European species is Cantharellus acervatus, the North American chanterelles represent a complex of distinct species,
such as Cantharellus formosus in the Pacific Northwest, Cantharellus roseocanis, and several distinct clades in California.
The phylogenetic distinction between Acervatus and the North American clades is highly relevant. Because these populations have been genetically isolated for millions of years, their secondary metabolite profiles and specific protein sequences have drifted.
Then you could be allergic to one but not the other.
It is entirely documented that an individual can possess a severe, violent, idiosyncratic reaction to the specific proteins in the European Cantharellus acervatus, yet consume the North American Cantharellus formosus without any adverse physiological response.
That is wild.
The immune and enzymatic receptors are that highly specific. A minor variation in an amino acid sequence or the structure of a carbohydrate chain is the difference between a gourmet meal and a clinical crisis.
Another prime example in the usual suspects category is the honey mushroom complex, the Armillariella or Armillaria genus. Again, geography dictates the specific taxonomy. In California, the primary species is Armillariella ostoye, characterized by a lighter orangish tan cap.
But in the Pacific Northwest, extending into the Rockies, foragers predominantly interact with Armillariella gallica,
which features a distinctively bulbous stipe base and a smoother cap. Both of these species are heavily harvested, and both are consistently flagged in toxicological reports for causing significant GI distress in a large percentage of the population.
The Armillariella complex presents a fascinating intersection of biochemistry and culinary tradition. The widespread reporting of GI distress associated with honey mushrooms indicates the presence of specific, highly concentrated secondary metabolites that act as broad-spectrum gastric irritants. They affect a much larger percentage of the population than pure idiosyncratic responses.
But there's a culinary trick to bypass it.
European mycological traditions have long recognized a specific chemical vulnerability in these compounds. It is standard practice to subject Armillariella species to a prolonged blanching process, boiling them vigorously and then entirely discarding the water before proceeding with standard culinary preparation.
The biochemical implication of that blanching process is incredibly revealing. It strongly suggests that the primary toxicological agents within the Armillariella tissue are both highly water-soluble and heat-labile. By forcing the tissue through that initial boiling phase and discarding the solvent, you are actively leaching the volatile irritants out of the chitinous matrix.
It's essentially a chemical extraction in the kitchen.
Moving further into the absolute requirements of thermal processing, the reports extensively cover the toxicological realities of morels, the Morchella genus, and the birch-associated boletes in the Lesinum genus.
With Morchella species, we are transitioning away from localized idiosyncratic reactions and looking at predictable, universally toxic compounds.
Morels should never be eaten raw.
Never. The literature is unequivocal. True morels contain varying concentrations of volatile hydrazine derivatives. While these compounds differ structurally from the highly lethal gyrometrin found in false morels, they remain potent toxins. If the tissue is not subjected to sustained thermal processing, these hydrazine compounds remain active.
And there's a really stark clinical example of this.
The clinical data from a notable mass poisoning event in Vancouver perfectly illustrates this. Raw morels were incorporated into a salad and served at a large banquet. Out of 483 attendees who consumed the raw tissue, 77 individuals suffered severe acute gastrointestinal distress, with some exhibiting subsequent neurological symptoms.
77 people at one banquet. That is not a failure of individual enzymatic pathways. It is a failure to chemically neutralize a potent natural toxin.
The requirement for thorough cooking also applies rigidly to the Leccinum genus. These are the distinctive boletes with the heavily scabrous stipes frequently found in obligate mycorrhizal association with Betula birch trees.
The toxicological databases show a massive concentration of poisoning reports linked to Leccinum species, particularly originating from Colorado.
The geographic concentration in Colorado was primarily a statistical artifact driven by a highly organized local mycological society systematically compiling tox reports combined with widespread, easily accessible alpine birch habitats. However, the biochemical threat is entirely real.
It's not just a statistical blip.
Analysis by leading taxonomists, including Dr. Denis Desjardins, posits that North American Leccinum clades synthesize a specific, highly resilient secondary metabolite that acts as a potent mucosal irritant in the human GI tract. Unlike the highly volatile compounds in morels, this specific Leccinum irritant requires significant prolonged thermal energy to achieve molecular degradation.
So a quick sear isn't enough.
Standard, brief sautéing frequently fails to reach the necessary thermal threshold. This results in the intact irritant surviving the digestive process and directly attacking the gut lining.
We have to analyze one final clinical case before we conclude, because it perfectly synthesizes the two major themes of our deep dive: the hidden complexities of molecular taxonomy and the stark realities of human toxicology. The reports highlight the historical confusion surrounding a macrofungus known as Leucoagaricus leucothites.
This is the gray variant story.
Yes. Macroscopically, it presents as a pristine, white-capped species with pale lamellae, and it has long been documented in the literature as a reliable, safe edible. However, for decades, field guides published explicit warnings regarding a gray variant to this species that consistently triggered severe gastrointestinal toxicity.
The gray variant scenario is the ultimate indictment of relying solely on macroscopic observation. For decades, the mycological community operated under the assumption that they were observing a single, highly variable species, and that the gray coloration was merely a minor phenotypic expression, perhaps influenced by localized soil chemistry or UV exposure.
They assumed the associated toxicity was purely idiosyncratic, a random intolerance to a specific phenotypic variant.
But when modern phylogenetic tools were finally applied and the ITS sequences of the standard white form were aligned against the gray variant, the genetic data exposed the error.
The sequence data proved definitively that the gray variant was not a variant at all.
Correct. The molecular divergence was absolute. The gray variant represents a completely independent, genetically distinct evolutionary lineage. It was formally described and recognized as an independent species, Leucoagaricus barciae.
Taxonomy matters to your stomach.
And crucially, the clinical data confirms that Leucoagaricus barciae synthesizes a specific suite of toxic secondary metabolites that are entirely absent in Leucoagaricus leukocytes.
So the people getting sick weren't having an idiosyncratic reaction.
The individuals suffering severe GI crises weren't experiencing unpredictable idiosyncratic reactions to an edible mushroom. They were systematically poisoning themselves by consuming a toxic cryptic species that morphological taxonomy had failed to differentiate.
It is the definitive proof that resolving taxonomic phylogenies is not an abstract academic exercise. The precise genetic identity of these organisms has immediate, severe physiological consequences.
It absolutely does.
So what does this all mean? We have processed an immense amount of data today. We started by examining how genome skimming pipelines can extract vital molecular barcodes from heavily degraded, century-old museum specimens in New York, proving that morphological convergence has been lying to taxonomists for decades.
We analyzed the highly specialized adaptations of novel clades in the alpine regions of China, documenting extreme cytological features and volatile organic compounds functioning as ecological tools.
We reviewed the phylogenetic evidence that formally evicted a highly recognizable cespitose conifer specialist from its historical genus, exposing active cryptic speciation across North America. And finally, we detailed the severe biochemical realities of idiosyncratic toxicity, proving that the complex secondary metabolites of even universally praised culinary species can trigger violent, highly individualized physiological rejection based entirely on the specific enzymatic and immunological profile of the consumer.
The overarching synthesis of all this data points to a singular reality. Biological systems consistently defy simplified categorization. The human desire to construct rigid, binary frameworks to categorically label a clade based on its pileal structure, or to permanently classify a species as universally edible, is fundamentally incompatible with the extreme plasticity and complexity of the natural world.
It's never as simple as we want it to be.
Whether we are analyzing rampant, polyphily driven by intense environmental pressures, or the highly individualized, mathematically complex interactions between fungal proteins and the human microbiome, the underlying systems are always more intricate than our initial observations suggest.
The transition to molecular phylogenetics and advanced biochemical analysis is finally allowing us to perceive the true shifting architecture of these relationships. I want to leave you with a final analytical concept to mull over. We rely heavily on codified texts, field guides, and taxonomic keys to confidently tell us where one specific evolutionary lineage ends and exactly how that organism will interact with our physiology.
We want definitive answers.
We do, but if the true phylogenetic identity of a macrofungus is encoded in microscopic ITS regions that frequently contradict its gross morphology, and if its ultimate culinary safety is dictated not by a universal constant, but by the highly specific invisible enzymatic lottery of your personal gut microbiome, at what point does the designation of edible cease to be a biological fact and instead become an ongoing, highly volatile biochemical negotiation? Next time you see a mushroom in the woods or on your plate, ask yourself, who is really adapting to whom?